BioCentury this week is brought to you by Jeito Capital, a leading global independent private equity fund with a patient benefit-driven approach aimed at financing and accelerating the development of groundbreaking medical innovation. Jeito's unique investment strategy combines significant capital and collective expertise to support biopharma companies and their management teams, ranging from clinical development to market access for cutting-edge innovations, with one main objective: go faster for the patient. Learn more at jeito.life.
Marty Makary's hold on the top job at FDA is slipping. We'll take a look at where the FDA commissioner stands and potential successors. Plus, solving AAVs' redosing problem could change the economics of rare disease gene therapy and fire back up industry interest in a modality that has fallen out of favor. We'll discuss presentations at this year's American Society of Gene and Cell Therapy that seek to tackle this issue. I'm Jeff Cranmer, host of the BioCentury This Week podcast, and joining me to discuss all of this are Editor-in-Chief Simone Fishburne and Steve Usdin, our man in Washington, both just back from our European conference, and Lauren Martz, Executive Director of Biopharma Intelligence, and my fellow Executive Editor, Selena Koch. Simone, Steve, Prague, is it as cool as they say it is?
No, it's cooler.
Oh.
Prague is an awesome city, in part because it's a place that's risen from, I wouldn't say from the ashes, but, if you compare it to pre-Velvet Revolution to now, there's very few places in the world that have gone from being so grim to being so joyful in such a short period of time. So, yeah, it's
So I could
awesome.
talk all day about Prague. I'm now deeply steeped in all these Prague writers from the little Prague bookshops. And yeah, there is so much history, even 100 years of history there that it really wears it on every corner and everybody. So it's a great city. It's a really pretty city. Will say, I don't know if I'm allowed to say this, but the conference center is not in the prettiest part of town. So if that's your only experience, go further into the old town.
Which is probably good, because you wouldn't want to have the old town. You wouldn't want to have that there. But no, but it's an awesome city, and the Bioequity Europe conference was awesome. Every single conversation, every single panel was a variation on China, AI, what the hell's going on at FDA.
One of my favorite things, this was just before the podcast. If you listen to our Bioequity podcast, you will hear this. Somebody said to me, if I had $1 for every time somebody said China, I could finance my new funding round, my next funding round. There's a lot of China
excellent
talk, but also a lot of energy. People, this is not a sleepy conference. This is a conference that people are really, really hard to be at. So.
And the interesting thing was, and I think this is different from Europe than the United States, is the talk about China wasn't like it is in the United States, that, oh, they're eating our lunch, everything's gloom and doom, or what are we going to do about it? It was really, how are we going to integrate China into global development, and how is that going to, how could that actually be a force multiplier for European companies that are engaging with China? That was really interesting. Yeah.
That's excellent to hear, as we are working on our program for the China Healthcare Summit, which will be in early November in Shanghai, 13th edition of the conference. Putting it on once again with Bay Helix and McKinsey. Hopefully that wets your whistle to come join us in China. And the talk with Pazder went well, Steve? I know that was a big highlight of the conference.
Oh, yeah. Yeah, we did the fireside chat, or the pub chat, or whatever you want to call it, with Dr. Pazder, and he was as provocative and interesting as ever.
Excellent. Well, as Simone said, we'll have a Bioequity Europe podcast recorded on stage in Prague with my fellow host, Steve Hansen. That will be out later this week. We'll also have on our sister podcast, Steve's conversation with Karen Knudsen of the Parker Institute. All right, Steve, Friday afternoons in the Trump administration. Always interesting. So we got word Wall Street Journal breaking the story that Marty Makary is on thin ice. Bring us up to speed, Steve.
So, yeah. So, people listen to this podcast frequently, read by a century, won't be terribly surprised to know that Makary is on thin ice. But I think it's important to say, what is it that we know and what don't we know? So I've spoken with people who are in touch with HHS Secretary Robert F. Kennedy Jr., with Chris Klump, who's essentially running HHS on a day-to-day basis, and other senior HHS officials. They've been looking for ways to push Makary out for months. So that's not really new. Makary wasn't Kennedy's choice for the job. Kennedy was never a fan of Vinay Prasad's. And there's a lot of reasons for the HHS displeasure with Makary. It centers on the chaos and leadership churn at FDA. Makary's frequent appearances in the media, including social media, aren't doing him any favors with HHS. What every HHS secretary and every president wants is a set it and forget it FDA commissioner. They don't want the commissioner in the news a lot because it's often bad news, and it's almost always a controversy that doesn't win any votes. I don't think there's very many voters that wake up, read about the real-time review program or the plausible mechanism pathway and say, well, I want to go out and vote for Republicans in the midterms because of that, So maybe the administration thinks that they're going to gain some support by talking about food coloring and food additives, things like that. But those are things the HHS secretary can do without the FDA commissioner opining on them. The reporting in the New York Times and the Wall Street Journal did kind of break this story on Friday. As I said, it revealed what BioCentury readers and listeners have known for a long time. There's this kind of angst about FDA and about Makary. From the perspective of the life sciences, FDA's rejected and dealt setbacks to a string of therapies, mostly but not entirely for rare diseases. A lot of its decision-making seems to be arbitrary. The policies that have been announced, the Commissioner's National Priority Voucher Program, the real-time reviews, were not carefully considered, and they're likely to have unintended negative consequences. Some aspects of them may not even be legal. FDA's lost about 20% of its staff over the last year. Makary said he has plans to hire 3,000 reviewers, but there's no evidence that he's even reversed the continuing attrition. So that's all on one side of the equation. And then on the other side of the equation, there's the reality that this isn't really up to, this being Makary's future, isn't up to Kennedy or Klump or the people at HHS who are trying to get him pushed out. It's up to Donald Trump. If you read the Wall Street Journal story carefully, it says that Trump had okayed a plan to oust Makary. No one, and I think probably even including Trump, knows exactly what that means. I think what it means is that, as I wrote on Friday, it means that Makary's going to go. That isn't really much of a doubt, but nobody has any idea of when it is going to happen. I actually think that all of this publicity that's happened over the last few days may delay his departure. I'm not sure, but I think it may delay his departure because I think that the plan was to give him a kind of a soft exit. It wasn't for him to leave with a cloud over his head. And all of this publicity makes that more difficult to achieve.
Hey, Steve, when I think about this, I take your point that it might actually delay it because that's the quirk of this administration or every administration. They like to set the tone rather than be seen to be reactive. But all that aside, when I think about Makary, when I read the things you've written and other people, in a way, there's a curious upside to this in that what I think seems to have brought him down is that he's only ever acted politically. And he's been, subject to being pulled in this direction politically and that direction politically. He's gone with the wind, he's gone with his gut. But all the way along, this just seems to have been clouded by political considerations rather than the science. And, when I say there's an upside, I mean, it's actually good that that's not working out, is what I'm trying to say. I think that you talked about a previous commissioner who very early on was faced with a political kind of interference and said, I have to clamp down on this now, otherwise it's going to plague my entire tenure.
Yeah, so what I've heard from other commissioners, I don't know Makary, but I've known every other commissioner going back to Mark McClellan, and they all say a variation of the same thing, which is that if you let the White House you let politicals in the administration think that they can push you around on one thing, there's never going to be an end to it. They're going to push you around on everything.
And that's the story of this.
And that's the story of this. And it's not so much the kind of FDA commissioner preserving their power or their perquisites or something like that. It's really about the effect on public health of having political appointees who aren't public health experts making decisions and influencing decisions in ways that may be well-meaning, may be intended to satisfy political constituencies, but aren't likely to result in decision-making that's the best for the American people.
All right, very quickly, you're going to tell everybody who's next in line, who the candidates are? I
have no idea. I don't think I've heard various rumors, but they're only rumors about who might be the next commissioner. But what is fairly certain is who's going to be the acting commissioner. So there's likely to be an acting commissioner. It could be for some time, depending on what happens with the composition of the Senate, after Makary leaves, whenever he goes. And again, we don't know when he's going to go. But after he goes, there's going to be an acting commissioner. There's actually pretty tight legal criteria for who can be the acting commissioner. And there's also a strong incentive for the Trump administration to adhere to those criteria, because if they name somebody to be acting commissioner who doesn't comply with the parameters of the law, then any decisions that commissioner makes could be determined to be null and void. So I don't think they're going to do that. The most likely course of action is to promote somebody from within as the acting commissioner. There's 3 people who would be qualified and who seem to be in the running. Grace Graham, who's the Deputy Commissioner for Policy, Legislation, and International Affairs. Lowell Stata, the Deputy Commissioner for Strategic Initiatives. And Kyle Diamantis, who's the Deputy Commissioner for Human Foods. I've heard that the most likely interim or acting commissioner would be Kyle Diamantis, but any one of those 3 could get it. It's also possible there's a list of people who are outside of FDA, who are appointees, senior appointees at other parts of HHS, who could get the job. In the past, twice, NCI directors have gotten that job on an acting basis, and one, Andrew von Eschenbach, was converted to get it on a permanent basis.
And if you're curious about who some of the other candidates are, Tracy Beth Hoeg, Sean Kvenny, and others, Steve has a tidy table in his story listing everyone that has a shot and could be the next acting FDA commissioner, should all this come to pass.
And I'm sorry, just one final point, which is that there's a tradition at FDA of having FDA commissioners who have served in that job for quite a long time and have been very important in policymaking.
All right, we're going to take a quick break, and we'll be back to talk AAVs and the American Society of Gene and Cell Therapy annual meeting.
This episode of BioCentury This Week is brought to you by the 3rd BioCentury Grand Rounds in Seattle. Advancing drug development requires more than discovery. It requires the right partners. The 3rd edition of BioCentury Grand Rounds US convenes venture capital, biopharma decision makers, and academic innovators in Seattle, June 3rd to 5th. This R&D-focused forum brings together leaders at the forefront of translational science to examine the breakthroughs, bottlenecks, and strategies shaping the future of drug and diagnostic development, and how to make early-stage R&D investable. Discover cutting-edge disease biology and platform technologies. Gain insights from emerging biotechs and academic pioneers. Schedule partnering meetings with VCs, pharma, and leading academics who can accelerate your path forward. Grand Rounds US is where rigorous science meets strategic capital, and where the right conversations move discovery toward development. Join us in Seattle and discover what's next in biopharma and who's driving it. Secure your spot. Register at biocenturygrandrounds.com.
All right, well, you heard it there, BioCentury Grand Rounds, right around the corner, but not too late to register. It is in, as you heard, the awesome city of Seattle, which has become something of a biotech innovation hotspot. And, well, podcast listeners, you're in luck. You can email conferences@biocentury.com or hit me up on LinkedIn, and we'll share a discount code with you. And that's good for delegates, presenting companies. Still a few spots left for presenting companies. And we also have posters. Simone, a word on the posters?
Okay. First of all, I have to say this conference is shaping up to be extremely interesting. Very hot conference. So we've got all these cutting edge topics that we're going to be discussing. Also, David Baker is going to give a keynote, and Mary Bronco is going to give a Horizon session. And I'm going to be interviewing Chris Arendt, the CSO of Takeda. So we have a lot of headliners there. But what I wanted actually to do with this podcast is I know that we have entrepreneurs and academic entrepreneurs who listen to us. And if they don't, go find one and tell them they should. But I do get people telling me that. Anyway, we would really love to see them. This is a conference for them. This is a conference where they can come and really present their work. We have poster presentations, which is just a great way to get your information and get your discoveries, even if they're really early stage, in front of VCs and farmers and people that we think of will be the people who one day partner with you or invest in you. And these posters have been really successful in the past. Very much encourage academic entrepreneurs or would-be entrepreneurs or wannabe entrepreneurs to come to the conference. And if they've got some cool work, send us a poster.
Yep, it's going to be a good time for sure. Well, over in Boston, other side of the country, the American Society of Gene and Cell Therapy is holding its annual meeting this week. The meeting is featuring multiple preclinical presentations that describe strategies to enable repeat dosing or overcome pre-existing immunity to AAV vectors. Lauren has been digging into this for a story that is out now on BioCentury. Lauren, what have you found?
Thanks, Jeff. So this is the second year in a row that we've looked into this issue of immunity against AAV vectors as a theme from the ASGCT meeting. And I think it's just because it's so important and because there really have been some big steps forward over the past year. Taking a step back, immunity against AAV vectors causes several different problems. It's obviously, a major contributor to the toxicity issues that we always are on the lookout for with all of these different AAV therapies. It's also, as you mentioned, behind two really big limiting factors for the applications of these gene therapies. The first is between 30 and 60% of people, any people, have pre-existing immunity to AAV vectors, which means they can't get an AAV gene therapy at this time at all. Even if you have a terrible rare disease and a gene therapy is developed for it, you're not eligible for it. And then there's also the issue of only being able to dose an AAV gene therapy one time. And if you think about the impact that has on just anyone trying to develop a gene therapy for a rare disease, it's really limiting, because once you have a gene therapy approved or a gene therapy in a late stage development for any indication, especially a rare disease, that's going to be used to treat many of the patients with the disease. So the market then begins to shrink. And if the gene therapy isn't durable, you can't dose it again. You can't help patients in the future. So we don't know how long some of these gene therapies are even going to last for patients. So it's a huge problem. And if it's solved, these are two slightly different issues that need to be solved. But if they are solved, you solve a lot of the financial disincentive that exists for developing AAV gene therapies.
And if I could jump in and just say, changing the economics for rare diseases is huge, because of course, in aggregate, they're not rare at all, even if any one of them is rare. But the conversation, I think, was something that drops out of it often or that gets ignored is the promise in prevalent diseases, right? There's this whole concept of vectorized biologics where you can encode an antibody or protein therapy as DNA sequence, give it as a gene therapy. So much wide open territory that could be traversed there if there was a way to do this. And then just, what was it, last week or recently on one of these podcasts, we were discussing how in vivo CAR T therapies, whether they were going to be successful was completely going to hinge on the durability of that first treatment, because these are what is an in vivo CAR T, but a gene therapy, right? They're delivered with a lentiviral vector. which can't be redosed. But what if it could? Back to you, Lauren.
Exactly. And so again, if we can solve these problems, I think it just bursts this field wide open. The biggest advance that we saw over the last year was maybe not even at this meeting, but we did hear about at ASGCT in the fall, a patient with a high enough level of neutralizing antibody titers against the AAV vector for a gene therapy in a Genethon trial for a disease called Crigler-Najjar syndrome was treated with this gene therapy. So what Genethon did was they treated the patient with an IgG depleting enzyme, imlifidase. This is approved for other indications. It's used in other settings, but there's been a lot of speculation that maybe if we bring down the IgG antibodies that pre-exist in these patients for long enough, you can treat them with an AAV gene therapy and it will be successful and reach its target, express the gene that you needed to express. And we will hear an update at ASGCT from Genethon on clinical data from that trial. Hopefully we'll see some additional information about how that's been working in that particular patient and what the next steps are, introducing this gene therapy for more patients who have pre-existing antibodies, maybe taking it to other indications. So that was sort of the big thing that happened. And then when you look at this meeting versus last year, there is a diverse set of strategies that are proposed to tackle this problem. So the IgG depleting enzymes are sort of the focus of this pre-existing antibody issue, pre-existing immunity. When you think about the problem of redosing, it's a little bit more complicated because you have there are more entry points for therapeutic intervention. So you want to prevent this immune response when you're giving the first dose. And then you want to deplete whatever immune response still does result. So you still probably have that need for depleting the IgG antibodies. But there's also a lot of work around how do we prevent those antibodies from being produced in the first place. Many different groups are proposing different strategies to deplete the B cell activation or, take out the B cells in general. There are some really extreme proposals like CAR Ts, which you have to kind of weigh the safety and benefits of using something like that. But different combinations of B cell depleting antibodies or B cell targeting antibodies are also proposed. And then there's another set of, this isn't a new idea, but as we're designing new AAV vectors, trying to design them in a way that evades the immune response has been a long-standing goal. There's some work around that. And then some more innovative ideas like cloaking the vector in extracellular vesicles, for example, that help it evade immune response from the start.
Lauren, I suppose my question is this. Your lead up to this, when you described the problems that are being addressed at the ASGCT conference, as you pointed out, those are long-standing questions, right? We've known about redosing being an issue. We've known about that for a while and the various limitations. And Selena, your sort of what if is obviously a really tantalizing one. What if you could solve this? And so what I want to ask you is whether you described now just several different strategies that companies and innovators, I guess they're not all companies, right, are taking to address it. Do you think that there's a sort of critical mass now? Do you think it's, what I like to think about is this ultimately an engineering problem? Is it something that you keep hammering away at it? You try different ways and the field is sort of incrementally getting to solve this. And I ask this because gene therapies, I don't know if it's more than any other, but it feels like it has just got this history of waves where they try it and then there's a disaster and it goes away. And sometimes it's a commercial reason and whatever. We're still knocking on the door really with gene therapies. But I think what I'm really asking is whether there's reason to believe that this massive activity that you're talking about can actually change the trajectory, start to solve some of these long-standing questions.
Are we at a tipping point?
Are we at a tipping point? There we go. Thanks, Selena.
So we have a history of identifying things maybe before we get to the tipping point. So we may be.
That's what we pay you the big bucks for.
Exactly. We may be almost getting to a tipping point. I will say, I don't think there's a massive amount of research around this topic. I think that it's something that people are working on. I think that companies and academic groups are chipping away at the problem. I think that what we've seen we have a patient with pre-existing immunity who was dosed with an AAV gene therapy, and we'll see how durable that effect was. I think that's a big step in the right direction.
Sorry, that patient would have been screened out of earlier trials?
Oh, that patient would have absolutely been screened out of earlier trials. I think there's still this challenge of preclinical models not translating to humans when you're talking about the immune system. Shocked to hear this, Lauren. Shocking problem, but
But Lauren, how much of the data would you say at this year's meeting is maybe in non-human primates, say, instead of mice or whatever? Are we seeing an advancement in that way?
There are some studies in non-human primates, which is, of course, a better model than a mouse model for this. But there's still, even with the clinical trials with gene therapies that we've seen, there are always unpredictable things that happen. So I think what we're expecting to happen is if you dose someone with pre-existing immunity with an AAV vector, and maybe the strategy that you've chosen doesn't work, hopefully it won't be effective. But we don't know what the risks are. We don't know how ethical it is to bring like a redosing strategy into humans. And I think it's just going to be a relatively slow process, but hopefully the first step is that you can treat patients with pre-existing immunity, and then you can use some of these B-cell depleting therapies that are becoming safer and more standard in inflammatory diseases and cancers as a way to sort of bridge this immunity problem. And maybe in the future, we'll have vectors that evade the immune system completely, But I think we're moving in the right direction.
So you list a bunch of targets in this story that people are trying, for the various, either to prevent new B-cell responses, address existing ones, address the T-cell component of it, so on and so forth. And whoever's listening to this can go read the story and see these various targets. But because there's lots of things being proposed and it's still early, does that mean right now would be a good time for the field to get together and think about how do we create standardized assays so we know how to use these things effectively in individual patients?
This would be a great time to do that. It's something that we talk about for a lot of different targets, a lot of different drug classes. Assays to measure neutralizing antibodies are not standard. They're all measuring different things from what I hear. You can read about it in the story. So it's hard to tell which of these approaches is working better at bringing down the antibodies, which is going to address the T-cell component, which is going to be probably going to be an issue when you come to redosing. So you sort of need to inhibit both of those processes. Yes, it would be wonderful if people came together to solve this problem.
Skeptical here. How many times have we said, if the industry would get together and solve this thing, they'd all do better from it? But
oh my gosh, I don't know, PD-1 might come to mind.
It's just, what about a single assay? As I said, they did it in COVID because they had to. So we know that they've got that muscle in there somewhere. Not holding my breath on that one, I'm afraid.
Just making an appeal yet again.
Well, if they do, then we'll definitely point it back to your comment on this podcast, Selena. We'll be like, maybe we can influence,
Lauren, I'm curious, are there any other things you're watching at the conference?
Always. Yes, there's a lot of interesting stuff within the abstracts that will be presented this week. So a few disease areas there are indications that stood out as hot to me this year. Muscular dystrophies, including but beyond DMD. There's a lot of research on limb-girdle muscular dystrophy, for example. I'm going to be looking into those. Gene therapies for the eye, of course, always a big topic. There's a lot of research into retinitis pigmentosa and the different genetic forms of that disease. Those are two things that stood out to me. Going back to Selena's comment on the encoded antibodies, one thing that I saw quite a bit of was strategies. Well, first of all, DNA encoded antibodies, vectors to create drug factories in the body. There's quite a lot of that. There are also different strategies to tune expression of and levels of the transgene and the protein that's produced from gene therapies, which I think will be increasingly important as you're talking about long-term production of therapeutic proteins and not just replacing a lost gene, for example. There's a lot of activity on in vivo CAR Ts. As always, there are a few new companies that are new to BioCentury in this list and a few different strategies to create the in vivo CAR Ts. yeah, just a lot of interesting topics within the abstracts.
Excellent. Well, I'll drop a link in the show notes to Lauren's story, as well as Steve's story on Marty Makary's tenuous grip on the FDA job. We'll see what happens there. Steve, of course, always working the phones, and Lauren always digging into the abstracts. That's how we roll here at BioCentury. Thanks for tuning in. Look out for our special BioEquity Europe episode this week, as well as the BioCentury show's interview with Karen Knudsen of the Parker Institute. And a thank you to Kendall Square Orchestra, which provides the music to BioCentury's podcasts.
BioCentury would like to thank Jeito Capital for its continued support of our BioCentury This Week podcast and our 26th annual BioEquity Europe Conference this May in Prague, Czech Republic.